This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 624, recorded on June 5th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody.
Happy birthday, Dixon.
Thank you very much.
Number 80.
Yep.
Wow, it's impressive.
That's right. Well,
you don't look a day older.
I don't feel a day older, actually.
Excellent. Also joining us from Austin, Texas, Rich Condit.
Hi, Dixon. Hi, Vincent. Happy birthday, Dixon.
Thank you, sir.
This is a wonderful milestone.
Yep.
It's 89 degrees here, headed for 93. Oof. Mostly sunny skies, and no chance of rain. And I do have to report to you that the forecast for this coming Tuesday is 106.
Wow.
Texas. That's Texas.
Texas. I'm living in Texas.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is 77, humidity is 74%. So it's 25C. And it's kind of cloudy and muggy and trying to think about rain.
Same here.
From Southeastern Michigan, Kathy Spindler.
Hi, everybody, and happy birthday, Dixon.
Thank you, Kathy.
Oh, and happy birthday.
Oh, thank you.
Here it's 84 Fahrenheit, 30 Celsius, completely sunny, no chance of rain.
Nice.
That sounds good.
From Madison, New Jersey, Brienne Barker.
Hi, everybody. I think I have the same weather as Alan, 77 Fahrenheit, and gray, and happy birthday, Dixon.
Thank you so much, Brienne.
So
appreciate it.
It's quite interesting that on this day, Dixon's birthday, where he turns 80, we're going to talk about sex and age and infectious diseases.
That's right. I've become an elder.
Well, Dixon, your part is the age part, okay?
Yes, just to be clear.
Okay.
So our guest from Baltimore, Maryland, Johns Hopkins School of Medicine, Sabra Klein. Welcome to TWiV.
Thank you so much, and happy birthday, Dixon. And I'm thrilled to be here. It took a lot to hold back from saying 80 and male. We'll be talking about that. It's 87 degrees here. It's sticky and humid in the Mid-Atlantic, and we are awaiting the birth of all our periodic cicadas in the area. So that just adds to the flavor in the Mid-Atlantic.
Baltimore in the summer. Yeah.
Every 13 to 17 years.
Wow, really? It's that cycle now?
Yeah.
Yeah.
Wow.
They're large and they're loud, and
you find them dead on the sidewalk outside.
Yeah.
When I graduated college, they almost canceled it.
Yeah.
And it was near you, Brienne. It was at just down the road from Drew. It was at the Madison campus of Fairleigh Dickinson.
Oh, they almost canceled the graduation, not the cicadas.
They did, because it was so noisy.
You can't cancel
hear
the cicadas.
When we got out of the car, we thought it didn't turn off.
Yeah, it can be quite intense.
Yeah.
It can be. It's exactly right.
So, Sabra,
it's a phenomenon of nature.
I've been wanting to get you on ever since, for a long time, but I saw you at that meeting in Baltimore, which was last summer, was it? The influenza meeting. I said, well, if you're ever in New York, and then of course after December, no one traveled anymore.
Absolutely.
But you said your mom is around here or something?
My sister and her family, 2 young boys, who she now homeschools in an apartment in New York City.
Wow.
Because of the pandemic. So no, I have not been on.
So I thought now we should just, since everyone's Zooming, and you, like everyone else, have been at home mostly, right?
I have. We've started, and I hope to share a little bit with you of some of the work we're doing with COVID-19.
You will.
So my lab is definitely active with the COVID-19 research, but like everybody else, we put everything in freezers and stopped all other research.
Yeah.
Wow.
So before we talk about sex and age and infectious disease and gender. Let's talk about your history. Tell us a little bit about yourself. Where are you from?
So I think being a mutt is really going to become a theme for me. So I am a military brat. My mother, who actually just a few weeks ago turned 81, is Irish Italian Catholic from San Francisco. And my father, who's a few years younger, Jewish from Manhattan. So you know, and there it was, the 60s. They found love in Europe while my mother was teaching on an Air Force base. And then I came along and they lived in Hawaii. I have no recollection of living in Hawaii. I do like to tell people that I am Hawaiian because I was born at Tripler Air Force Base. So my point is we moved around a lot. And my father ended up retiring from the Air Force in Virginia, so not too far away, still the Mid-Atlantic. And so I think I would say that I am from Virginia, and that's where I finished high school and went to college. And I really thought I was going to be a psychologist. And I was very interested in the brain and in behavior. And I actually thought I was going to become specifically a primatologist, that I was really going to, be the one to travel to exotic locations and study primate troops. And then I learned how tough that is, and that I would probably end up working in a zoo, not as easily in academia. So I switched to smaller animals and continued my love of the brain and behavior in psychology departments, because this was before we had all our, fancy neuroscience departments. A lot of that was coming out of psychology. So that's where I began to think about sex differences and hormones, and how hormones can result in very profound differences biologically between males and females. And I started thinking about it in terms of evolution and mate selection. And so that was a lot of my dissertation. What goes into female choice? Why do females choose the mates they do? And I started thinking about how infection may be one factor, that males are either advertising being able to overcome an infection, or they might be advertising that they're susceptible to an infection, and how do we use that information? So that's when I switched over and did my postdoc at the Johns Hopkins Bloomberg School of Public Health with Greg Glass. And I was doing a lot of my work in wild rat populations in Baltimore. So I was keeping the hantavirus world alive and well, trapping rats and looking at infection status and comparing males and females, and going into the lab, all BSL-3, and doing some behavioral studies to look and see, to what extent do females use this information in mate selection. And from that came this love of hormones and immunity and the male-female differences, which became very profound to me. And so I was I grew up a bit of a mutt, not really being defined by one place or one region of the United States, but having family everywhere. And I think my research sort of reflects that I switched from psychology and neuroscience into microbiology and immunology. And over time, and it took a while, I gained acceptance, and now here I am.
So you've been at Hopkins for a long time now, right?
Oh my goodness. I grew up there. I mean, if there's one place, because I didn't say it, but I got my PhD in psychology. I switched over to microbiology and immunology for my postdoc, and was promoted eventually to assistant professor under another wonderful virologist, Diane Griffin, who, always was very supportive and believed in my ideas, which at the time were obscure. And I just kind of came up the ranks, and in November of 2019 was promoted to full professor.
Great.
Congratulations.
Hey, thank you.
So do you sail?
I do not. I do not.
I must say that had you been a psychologist, you would have been a great one, because your voice is very soothing.
Oh, that's so kind, because I promise you that my daughters do not say that to me. I needed to hear that, because I've been in quarantine with 2 teenage girls.
Yeah, I can imagine. Yes.
Mm-hmm.
Yes.
Thank you.
Quarantine is not easy.
No.
All right. We're going to, as you hinted, we're going to talk about the effects of sex and gender and age on infectious disease. But I think it's important to start off by having you define sex and gender.
Yeah. So you'll appreciate this. Long ago, I gave a seminar at Colorado State University in their microbiology department. Charlie Kalisher was the one who invited me. And the only thing he said when he introduced me was, this is Sabra Klein. He said he liked what I do. I make him think. And that I've taught him that sex is who we are, not what we do. And then he just stepped off stage and left me there.
Charlie, that's Charlie.
That's Charlie. So we'll be talking about the sex of that defines who you are, not what we do.
It's a noun, not a verb.
Thank you. Well said. And we define it based on our sex chromosome complement. So for most of us, though not all of us, we either have two X chromosomes, which in mammals would be female, or we have an X and a Y chromosome, which for mammals would be male. We also define it by our reproductive tissues. So whether you have testes or ovaries, testes being male, ovaries being female, testes that are going to secrete higher concentrations of androgens like testosterone, and ovaries that, at least during our reproductive years, are going to secrete high concentrations of estrogen and progesterone. It's not to say females don't have androgens. We absolutely do. We have circulating concentrations of them, but they are significantly lower than males. So one of my pet peeves is to be told that testosterone is a male hormone or that estrogen is a female hormone. For all of you, going back to the brain and behavior, for the males on this podcast, any sexual differentiation of your brain that occurred early in utero, that was all based on estrogen. So estrogen, these things play roles. It's just in the relative concentration. Mm-hmm. And I think you want me to kind of distinguish that from gender, which I think historically we all like to use the word gender. It always sounded a bit more politically correct than to say the word sex.
Nicer in polite company.
Isn't it? It doesn't raise as many eyebrows, but it resulted in the phrase being used incorrectly. And gender we often refer to, and I'm not talking about gender identity. I mean, that's something separate. But for gender and gender differences, when we're thinking about males and females and the comparisons of males and females, this is really about the social, the cultural norms that maybe define both who we are, but what we do, and in some countries, what we may even have access to. So when I think about infectious diseases, if you think about 70% of the healthcare workers, those frontline workers that we've been talking about and celebrating a lot, it turns out 70% are female. And so exposures associated with that, they may be gendered, they may be gender specific. You may have greater risk of exposure for women because you are going to have more women on the front line. And that may not have to do with their biology. It just may have to do that it's putting them in a place. We see the same thing if we're going to talk about flu or other pathogens that we get concerned about that kids give. A majority of teachers, a majority of elementary school teachers are women. So you have greater exposure. So, and sometimes I think, especially if we get to talking about some of the epidemiological data for COVID-19, dissecting out what's our biology from what's our behavior and societal norms, I think becomes increasingly difficult. That's when I like models, they let me make it a little easier to study the biology because I am a biologist at heart.
Yeah, and I think we've got a running pet peeve on the show about papers where people use gender to mean sex.
Yeah.
And that has come up a few times.
And I've heard it. And,
yes, good.
And just, it warmed my heart.
Good.
So have we known for a long time that that the responses to infectious agents and the actual disease is controlled by sex and gender?
We have. I can I can go back. The earliest documentation that I have, and there might be earlier, and this is when probably your listeners become this wonderful resource because they will probably be able to prove me wrong. 1926, there was a beautiful study of tuberculosis. And really showing very distinct patterns of, what we might say are male-female differences in deaths from tuberculosis being greater for males than females. And, what I loved was in this paper, which was published in what was then the Journal of Hygiene, it even went so far as to talk about differences in clothing, because at the time women were wearing corsets. So there were some concerns about, or questions about what does that constriction do to susceptibility. We didn't know about sex hormones yet. Those weren't discovered until the 1930s. But the article gives some interesting speculation that there could be factors in our blood that are impacting, why men and women may be responding so differently to infectious diseases. So I think there are pockets of knowing about this for bacteria, for viruses, for parasites, even for fungi. But they were often, just, the observation like, oh, and by the way, we did notice this, but it wasn't something people were really studying and trying to better understand and thinking about therapeutically.
It's always great when you're reading one of those old papers and they say something like that, like we think it might be something in the blood. And you're like, you're so close.
Like, oh my gosh, it's like turn the page.
Right. It's like reading the old papers about the transforming principle, and somehow it's yes, you've almost got it.
Oh, it is. It is.
So I worked with worms for a long time, and the worm that I worked with, Trichinella, it would beautifully infect male mice, 3-month-old male mice, right? There's no variation. The standard deviation for each mouse was very low compared to the bunch. Female mice, forget about it. So you end up working with the males just because you get a more even result. And
you get a phenotype to study.
And you do. That's true. So I wish I could go back now and do the females, because that would be much more interesting, I think.
I love it. And people have, but just as you described, I used to, especially in my days of doing hantavirus research, I would be at the American Society for Tropical Medicine and Hygiene meetings annually. I would go. I know. Well, I would attend all the worm talks, and I'd ask that question, and then I'd be told, well, because females, we really don't get much of a phenotype, and I'd say, but maybe therapeutically, there's something really interesting. Why how is it that these worms, they don't even need to, they can't even complete their life cycle.
Exactly.
In these females.
That's right.
And
I've always found the females more interesting anyway.
Oh, thank
you. So obviously these differences are not just humans, but mice, we've mentioned, many other animals, I assume, as well, right?
Absolutely. Birds.
How about insects?
There's data in fish.
Insects?
Yeah. Insects. There are definitely data in insects. Yeah. There's some data from fruit flies and even mosquitoes.
All right.
I imagine risk factors from eating your mate would probably be factored in.
You like praying mantis?
Praying mantis. That I don't know.
Spiders do that all the time.
Yes.
Sabra, when you're working with the animal models, is there also any difficulty in dissecting the effects of sex and gender?
Yeah. There can be. And an example that I'll give you, most of us who work with mice, for example. Ooh, I am so sorry. I know. I'm ruining your podcast. I don't know what happened.
We can still hear you.
You're good.
All right.
What's wrong?
I don't know. Somehow Coldplay began playing in the background of my computer out of nowhere. Okay.
Is that your favorite band?
Sorry.
Is that your favorite?
Actually, it is. It really is. So I'm a big fan. I've written many a manuscript to Coldplay. Okay, so sex and gender in our animals. So anybody who works with mice knows that you are charged per diem. So that means for the food and the water and the HEPA filtration and the care of your animals, which are all, really good, nicely regulated, we pay per cage. And the maximum number of animals that we can house in a cage, 5.
That's right.
So if you're going to charge me, 60, I think we're starting to get closer to 70 cents a cage per day, I'm putting 5 mice in that cage. I'm not putting 1 and I'm not putting 2.
That's right.
And we and others have shown that when you want to look at some testosterone data and see error bars that are much larger than what was described for the worm studies, they're huge, and they really do play out nicely with the dominance hierarchies that are going on in those cages.
Exactly.
And, it plays out and it does affect the immune system. So I worked with some vets at Hopkins because I can't afford what the answer is. And then I worry, well, then if you're individually housing all the males but not the females, then you've got maybe a confound. But you get some aggression in the males, and they do have these hierarchies. The females, they work together much better. And so we bring in mice to our colonies. I don't breed at Hopkins, but we bring in mice to our colonies at 5 weeks of age. And that's before they've had their surge of testosterone. So they're still, they haven't yet gone through puberty. They're cute and playful. And what you do is you let them, while they're in those ages, form their social groups, and they tend to be a lot less aggressive with each other. So so that's one of the creative ways that vets at Johns Hopkins help me figure out a potential. It's not always the solution. Sometimes I still have that one male who beats up everybody. He's the bully, and we remove him. But that's how we deal with that. But I do think that would be an example of how.
As you talk about the animals, I'm extrapolating to humans constantly.
It's too easy to do. Yes.
No, we all do it.
It all makes sense.
We all know that guy. Yeah.
Right, but you have to add that female mice that give birth in the cage often eat their young. So that's another problem.
That is very true. And you want to get an aggressive animal, you want to see aggression. It's called maternal aggression. It's been very well studied, and the mechanisms that control that. Right. Try to put your hand in the cage of a female who just gave birth and is guarding her young.
So we had this cannibalism problem about a year ago. And the solution was to leave the male in the cage, actually, because usually you take them out after a while, right? But our vets here in Jackson Labs actually told us, leave the male, which the animal people don't want to do, because now you have a litter, the female and a male. It's too many, as you said. It's more than 5.
Yeah.
We had to apply.
The fire department will shut them down.
We had to apply for special permission.
Yeah.
But it works. It actually works.
Great. So I love that. I love when we learn these kind of tricks too. And I think this actually improves the quality of the research that we're all doing, and it cuts back on some of the variability.
Oh, absolutely. I first heard you talk at ASV a couple of years ago, and you gave this amazing talk. And I remember the whole time kind of wanting to go back to my PhD project and think more carefully about the sex of the mice that I was using, because I did a project where I was given a breeding pair. And I used the males because I wanted to use the females to be more breeders. Yeah. I didn't think about it any more than that.
I know.
And yet I realized how much more I could have gotten if I had been taking that into account at the time.
Well, and this is something that's now just recently, I think, been codified in the NIH guidelines, right?
Absolutely. Right. So Janine Clayton has really been a wonderful leader of the Office of Research on Women's Health at the NIH. And, and Francis Collins has been very, very supportive of this. And together they announced this concept of sex as a biological variable needing to be considered in all grant applications. That we do need to be thinking about that. But I think, much to the previous point, I think what ends up happening, though, is we can pay lip service and grants. And then when we publish papers, we can go back to saying mice were used. And so I also, I try and ASM and their family of journals have been very, very good about this, that at all primary research journals, they now have in their instructions for authors that you need to account for or just report the sex. So that people like myself who maybe just need to know, do my data match what others have shown in females or males or both?
Then there's the controversy about inbred versus outbred strains,
absolutely.
Right?
Absolutely.
Or wild-caught animals. You said you were working with wild rats for...
I was.
How do they compare with the domestic ones that we can pick up by the tails and not get bit?
Yeah, so those nice domestic animals that you get to work with. First of all, we are all studying obese animals. Ad lib means obese.
Yep.
My students who had done all work with me with wild-caught rats, the first time they cut open a laboratory rat, they thought that the rat was oozing pus. And
that's right.
Thank you. It's all of the subcutaneous abdominal kind of fat.
Exactly.
And then to cut in and see it in the peritoneal cavity as well. And you can't find organs. And I will tell you, the spleen of a wild-caught rat, it looks like a tenderloin steak. It is gigantic. It's gigantic. They're getting exposed to so much. And
so very few of us are inbred animals. So I guess that
I agree with you.
The outbred wild animal is the gold standard.
I agree with you. Well, I mean, and that's why there are some interesting groups, doing work along those lines with the pet store mice.
Yeah.
To get at that point.
Collaborative cross.
The collaborative cross. Yeah.
So you've done quite a bit of work with influenza viruses, both in people and in mice, right? Let's talk.
Yes.
We'll get to COVID-19 for sure, but let's talk about that to kind of set the stage for the mechanistic stuff. So tell us about that.
So flu has been very interesting. What we've shown, at least with influenza A viruses, both H1N1 as well as H3N2, and this has been primarily initially this, was in mouse models, and then I'll get to, what we have done and looked at with people. Adult females were consistently more susceptible, depending on your dose. This could result in mortality, but we often like to work with lower doses. Most of us, especially young adults, are not dying from flu, so we tend to work with lower doses. And so what you find is that females just get sicker. They lose more weight. Mice become hypothermic when they get really sick. So unlike the fevers that we get, they lose body temperature. They get real cold. And they show just all kinds of, we worked with our vets to really define some of the clinical disease that we see in our animals. So we went on to show that this was really immune mediated, not necessarily viral mediated. So this wasn't about females not being able to control virus replication in the respiratory tract. And this wasn't about females not being able to mount an adequate type 1 interferon response. We really showed that all of that was intact and really equal to that of the males. But what we ended up showing, both when we looked at, if you will, the cytokine storm, which I think everybody talks about these days, as well as if we looked at pulmonary pathology, and also working with a physiologist at Johns Hopkins, Wayne Mintzner. We've actually even looked at pulmonary function. So the real physiology of the lung, really getting into gas exchange and things like that. All of this was so much worse for females. They had more cytokines, they had more pathology, they had worse pulmonary function, and for a longer period of time. And we really showed that this was inflammation based.
So, and do you know what's doing? I mean, is that a hormonal effect?
It is, yeah. So it does seem to be hormonal. I think importantly, what we've gone on to show, and we have something in press right now, a nice paper in press at PLOS Pathogens right now, really kind of outlining how testosterone and really signaling of testosterone through the androgen receptor in the lungs is critical for dampening that inflammation and providing that protection. And so for flu, this seemed to be really protective effective for males to have this dampening of inflammation. But, going back to the worm story and for some other pathogens, that dampening of an immune response, and especially an innate immune response, sets males up to often be more susceptible. So that's when I started to come up with ideas that, well, maybe when the pathology is being caused by inflammation, it might be females that suffer a worse outcome. And alternatively, when the pathology is being caused by an inability to control virus replication, it may actually be males that are suffering a worse outcome. And so that's like what we see both in mouse studies as well as human studies for things like hepatitis C. They can't control the infection. You get greater virus replication and inability to clear the virus. And then this leads to longer term damage, in the case of hepatitis C, to the liver. That you see in males. So, I do think we really have to pay mind to the etiology when trying to better understand these male-female differences.
So in your mice, can you give the females testosterone?
I can. Yeah, it works. It works. They fight more, and that actually is true. But it works. Okay. They do better with the flu infection.
How about
this really kind of highlights the delicate balance, right? I mean, you've got just based on the sex hormones, if you're a little bit to one side or a little bit to the other side, well, you get a worse outcome with this virus, you get a better outcome with that virus. And is this just evolutionarily, is there any benefit to having the two sexes segregate this way, or is this just a byproduct of the fact that testosterone has a bunch of functions and estrogen has a bunch of functions, and they have these side effects?
I would argue it's a byproduct. I think you're exactly correct. I think evolution is favoring anything that leads to increased reproductive success. And so if it's going to increase my ability to successfully carry children to term, and those hormones, I couldn't do it without them. That's fantastic. But you know what? If down the road it makes me more likely, and I'm 80% of all autoimmune disease patients are women. I have two. All women on my dad's side of the family have autoimmune diseases. But who cares? We've had our babies. Now if I can just get them to live and reproduce, who cares that my joints suffer?
What happens after that? Yes,
that's exactly.
I take exception to that.
I know. Nobody likes this, but it is true.
We're the institutional memory. We can help nurture the grandchildren. And
those are called helpers at the nest. Yes, helpers at the nest are needed.
That's right.
They ensure the reproductive success of subsequent generations.
I was going to ask a question about the autoimmunity that you've already mentioned, but I had one other question, and I don't know if your experimental setup even would let you measure this. Okay. But do you see any differences in acquisition of infection?
Oh, that's such a good question. Yes. So we've not seen that. Well, how's this? That's been almost impossible for us to study in the context of flu. Because as you know, it's notoriously underreported. So it's been really challenging to try to get at that in the context of human. And as you may or may not know, mice are terrible at transmitting respiratory infections because they don't have the cough reflex. So it's very hard to get what's in the respiratory tract back up. But where your question is really interesting is about sexually transmitted diseases. And that's where I think this can become very interesting. And so, but then you get into our anatomy. So now we're going to start to get close to the sex of what we do, not who we are. But our anatomy differences absolutely can play a very important role in that acquisition. But there are people who have done some interesting studies in primates with SIV. And what they have shown is that if you do anal exposures, so there you're controlling for the anatomical differences, you actually do get higher viral loads in males as compared with females. And I make the point that it was a model because then you can also control for the dose and the timing and so many of the other things that can be complicated.
So is it fair to generalize that the immune response to infections or the immune system in females is different than in males? Is that fair?
I think that's fair.
So why? Is there any, in the context of evolution or childbearing or whatever, does this make sense to you?
Absolutely. So, it turns out that, every immune cell in our body has receptors for our hormones. And receptors, these hormone receptors, especially for our sex steroids, these are kind of intracellular receptors that when bound to their hormone ligand translocate to the nucleus and can bind to a part of the DNA where you have a hormone response element. So they can actually transcriptionally regulate gene expression in our cells. And so that is what I think they do. I think they're modulating and they're doing a pretty decent job at times of turning on or turning off. And a lot of this can change, and this can change over our life course as concentrations of our hormones change. But I will say that, I think as as the omics revolution continues, there's a growing body of literature to suggest that both genetic and epigenetic regulation of gene expression, some of which is completely independent of hormones, and some of which has been reported to occur in kids under 5, where we wouldn't necessarily think about prepubertal as being a time where hormonal regulation of an immune response would be significant. We're seeing these differences, and myself and others have focused in on X-linked genes, just as maybe just a starting point. But more and more, we're finding that even in the majority of the shared genes that all of us on this call have, you see differential regulation. And I think we're going to really start to, understand a lot more about that epigenetic regulation. And, I think the people leading a lot of this are, in the autoimmune disease literature, because there's just, everybody wants to understand, why are most autoimmune disease patients women?
So go ahead, Brianne. Go ahead.
Does it seem like there are, I know of one example, but I don't know if there are others, of many immune-related genes that escape X inactivation that might be part of that?
Yeah. So I think there's a growing list. So the one that I've kind of honed in on and studied the most is toll-like receptor 7. And what turned me on to that, obviously, I saw everybody's eyes light up. It's like, you say TLR7, it's viruses, But in the autoimmune disease literature, there's also a lot of interest, and they had shown escape from X inactivation. And so for just all your listeners, generally speaking, just because something is X-linked does not mean that females should have greater expression through what should be, or what the textbooks tell us should be, a random process of X inactivation. One copy that we've either inherited from our mother or from our father should be inactivated. But some genes, and we and others, people just keep consistently showing that TLR7 just escapes this. Another one that's kind of interesting that seems to maybe escape or show higher expression, and again, some of this can be epigenetic, is FOXP3, which is another one. And then CD40 ligand is another one that has been studied. So it's very interesting that once people really start delving deep methodologically into these genes, both in humans as well as in animal models, you really start to see some differences. And, going back to your comment about being outbred, I love, and I think I just love this word and this phrase that it was Barbara Maijan, who's a geneticist at Johns Hopkins, who coined the term, or at least I attribute this to her, right or wrong, that females are a mosaic, that we end up being this mosaic of expression of these genes from both our mothers and our fathers, whereas males, especially for those X-linked genes, you only inherit your X-linked genes from your mother. And so what does that mosaicism and that diversity do?
So you triggered a thought in my mind as you were talking about the males when they get together. There's a dominance, obviously, and you see the bite marks on all the mice except one.
Yes.
Primates, except for us and most other mammals, have a dominant male.
Yes.
And a harem.
Yes.
All right.
Absolutely.
Gorillas, bonobos are an exception.
They
bonobos
are an exception.
Are a big exception.
Yeah.
But chimpanzees, they're
my
vicious.
goodness, vicious.
Right. So where did we lose that? What happened to humans?
Did we?
Didn't.
We?
What I mean is, if you look around the world, you usually got one male and one female living in the same domain, right? When you go into the wild, you've got one male and you've got 20 females.
But maybe this starts to become about, philosophically about what our social norms are, because there are always ongoing debates about, are humans really monogamous?
I know. Well,
and so,
we're not a herd animal.
True, but that can also be social norms.
So what happens if the dominant male in a wild population gets sick?
Oh, that's such a good question. So there have been studies that have done that.
The whole dynamic of what happens next, right?
Absolutely. And there are, and this is where, data get kind of messy, because there are definitely data, and there were a lot of studies that were done taking blood smears or looking in feces in wild troops of animals to get at this exact question. And, it was referred to in the 90s as the immunocompetence hypothesis is the downside of having higher testosterone and being that dominant male and maintaining that territory, that you're more susceptible to infectious diseases. And, there are some data to show that you do, it seems to have a higher parasite load. And so maybe what you're also advertising to a female is, I can withstand this and still look good and still maintain, my territory and my food supply.
That's right.
And everything else that I can provide, not only to you, but to your offspring.
Right.
So
flu starts in wild animals, right?
Absolutely. And I don't know if there's a sex difference.
You've got that dynamic first, and then you have a transfer into the human population.
That's exactly.
That's a fascinating connection, actually.
There's some people starting to think about, these differences in the reservoir populations of a lot of these zoonotic pathogens. And that is what I was doing. And I was showing that for a zoonotic pathogen, these hantaviruses that are carried by wild rodents, it's predominantly shed and transmitted by males. And they are shedding a lot more of it in their saliva, and they shed it when they're fighting. And it's not that females can't get infected. Going back to that question about acquisition, they absolutely can. They just don't get exposed to it as much because they are in the burrow, which turns out to be cleaner than you would think. Not as, I think it's kind of embarrassing to say that I maybe caught well over 1,000 wild caught rats in my time, but I took them off the streets of Baltimore, so maybe that's a good thing. Yes. They are so clean. No ectoparasites. They're so clean.
So that makes me, we just had Peter Daszak on the other day.
Oh, okay.
Trapping bats, and I bet they never look at the sex of the bat.
No, but they, but definitely some of the bat people, Tony Shounts and others have commented that, they do keep track, and they'll need to look.
Well, they have samples they take a little punch biopsy of the bat so they could figure it out. It would be very interesting to know.
I agree.
Right?
I agree. Because then it tells you maybe a little bit about some of the factors that contribute to making an animal maybe a super spreader or a more superior reservoir. I think we often assume that in these animal populations, all animals are equal. But there is some diversity, and we had seen that.
No, he was telling us that, only a low percentage of the entire bat population is infected at any given time. It's cyclical, right? And who knows what controls that? That would be
absolutely
really interesting to parse out. So I wanted to ask you about influenza. You mentioned in mice, the females get more serious disease. So what happens in people?
So in people, we were commissioned by the WHO during the 2009 H1N1 pandemic to work with them and analyze some of these data. And what we uncovered in that report was that, similar to what we were seeing in mice, and I like talking about the 2009 pandemic is because it was a novel flu virus, because data gets very complicated with flu because of all our pre-existing immunity, which we can definitely, talk about, especially in the context of vaccination. But for the 2009 flu pandemic, for those countries that were parsing data, and the countries that, I'd love to call out as just being the best at analyzing data through what has often been referred to as a gender lens. I think it should be a sex and gender lens, but I'm not in policy. Canada, Australia, Japan, and what they consistently showed was that females in their reproductive years, you had greater hospitalization rates. So more hospitalization of women during the reproductive years. In later ages, it was males, and at younger ages, it was males.
I do remember that pregnant women were particularly susceptible, right?
Right. And they were prioritized when the vaccine came about, I think.
Right. Yeah. Oh, they absolutely were. Pregnant women and healthcare workers. And, I think, for someone like myself to actually see that sex-specific recommendation come out that we needed to prioritize pregnant women, there were data from previous pandemics, including the 1918 pandemic. We knew pregnant women would fare worse. But sometimes these things, history gets lost. We like to repeat history. So that was really exciting for me. I will tell you that, more detailed analyses that occurred in Canada went on to show that even if you removed the pregnant women from the analyses and just compared non-pregnant women to men, you still had more hospitalizations. So, there was also, in addition to pregnancy, just something about being female, which, I would argue our mouse studies suggest could have something to do with inflammation and that inflammatory response to the novel virus.
Do you think it's a testosterone-based effect also in humans?
Yes, I do.
Would that be you can go next. Would that be a therapy then?
It could be. I think what's tough, it is interesting. And again, we're seeing this play out now in COVID-19 because this is all being thought about therapeutically, which I did not think I would see in my lifetime. Because going back to, this conversation that all of you, brought us into about evolution. When you're messing with our sex steroids, you're messing with reproduction, you're messing with fertility. But this concept, so I would argue though, yes, this concept that maybe you could give these things in a very acute way, not long-term. And flu is acute. I think you could have been doing some of that and down-regulating some of that inflammation.
So this is a good segue into my question, which is in these human studies, is there any accounting for whether or not females are taking oral contraceptives?
Yeah.
And does that have any effect?
Oh, I think that completely has effects. No, so it's something that, so I am a part of the Center of Excellence in Influenza Research and Surveillance at Johns Hopkins, led by our fearless leaders, Andy Pekosh and Rich Rothman. And, they've embraced these things. So we are accounting for this. You need multi-years of data to really start to tease this apart. So we're not quite there yet, but I do think this could matter. I will tell you that also through my participation in what's referred to as SEERS, the NIH gave us access to plasma samples from some of the safety studies that were conducted with that 2009 H1N1 monovalent vaccine that was recommended first and foremost to pregnant women. And we compared men and women, and we compared them in their response, both their immune responses, but also their sex steroid concentrations, and to look at the association, both in reproductive years, so 18 to 49 years of age, roughly, and then individuals 65 years and older. And what we found really clearly was that sex steroid hormone concentrations were associated with the immune response to the flu vaccine in these people. So for women having higher estrogen concentrations in both young and older women was associated with greater antibody responses. And in men, at least among the younger aged men, 18 to 49 years of age, higher testosterone was was significantly associated with lower antibody responses. Among older men, we lost any association with testosterone and the antibody response. And then, we can go to our animal models, and that's what we did. And we can manipulate the hormones. We can take out the testes and have some that have no testosterone and others that we give back. We can take out the ovaries, have some animals that have no hormone and others that we give back. And so that's how we can get at least some of the cause effect. I still don't know exactly which cell population. I mean, these are the things that we're doing. Is it directly on the B cells? Is this T cell help? I mean, these are the things that we're doing now.
So I was wondering about this with the animal models. You mentioned taking out the ovaries or taking out the testes. Can you do this at an early age, like pre-puberty?
Yeah.
Can you make a kind of a sort of a gender neutral animal? And
yes,
then
you can
manipulate them. Okay.
Absolutely. Yes, you can. We haven't done that, but those studies have been done in order to modify really other physiological systems, because it really turns out that, there are sex differences in the functioning of our kidneys. There are sex differences in our hearts. I mean, the Red Dress campaign is about being aware that heart disease strikes women differently than men. So these other physiological systems, they've done some of that.
So in that sort of experiment, could you, for example, remove testes from a male animal pre-puberty and administer female hormones and get a response that really closely mimics the female?
Absolutely. Yes, you could. Yes, you could. And it turns out, that the way we would go about doing that pre-pubertal time. So in utero, you have a surge of testosterone, the males on this call, and this is true in our rodent models. The timing of it differs because, I don't know that all your listeners know this, but mice are born significantly less developed than humans. So again, the timing and when this occurs during gestation is different between mice and humans. But the differentiation that occurs, so we all start out with this bipotential gonad that can kind of go either way. And disruptions can have it go another way or have it stay a little bit of both. That's intersex. But so you can do these manipulations often in utero and then really shift those responses.
Okay. The group of animals that I'm most fascinated with regards to that are fish.
Oh.
If you alter the temperature while the egg is developing, you get all males or you get all females.
Isn't that? I know.
The barramundi. Do you know the story about the barramundi in Australia?
I don't.
It's born a female.
Oh.
Then it migrates to the ocean and becomes a male, and comes back and mates with the females on the way out to become males.
Yeah. Right. Well, I mean, you're starting to get at the fluidity of some of this. And sometimes that gets lost because somehow for most of us, this all goes very well. And we are born either male or female, and that's the end of it. But then that concept of intersex or having an extra chromosome, so I will tell you, in individuals who have two X's and a Y, they're more likely to develop those autoimmune diseases, so going back to a little bit of maybe how we parse out some of the human data to speak to some of those genetic effects that I still think shouldn't be ignored.
One of the things I wanted to touch on was that you've used immunization. You've already mentioned this, but you've used immunization to study differences because it's kind of a nicely controlled environment, right?
I know.
But it's a different question because it's disease versus immune response.
Yeah.
And you just said that in mice, at least, testosterone is associated with a better antibody response, right?
Worse. Testosterone is a worse, both in humans and in mice.
Right, worse. And that may actually explain the lack of immunopathology as well, right?
Yeah, absolutely. Yes.
Okay. But anyway, tell us something about vaccine responses and how that tracks with sex. I don't know if you know about gender and age too, of course, you're interested.
Absolutely. So in terms of, your first comment about sex, what we and others have consistently shown is that females tend to mount more robust, greater antibody responses than males. In the context of flu vaccination, we've been focused on trying to show this not only in terms of the quantity, but the quality of that antibody response. So, and then again, our mouse studies, we can then go and challenge. And I realize human challenge studies, they're out there. We've not, done that. But when we challenge our mice, we've been able to show that females are better protected. Mm-hmm. So there really is some efficacy to this. And as you noted, we've shown that both sex hormones, but also going back to that escape from X inactivation, we've also shown that toll-like receptor 7 is intimately involved in this as well. Why females have greater, in this case, antibody responses, antibody-dependent immunity following vaccination. Does
the flu vaccine efficacy differ in men and in terms of protection?
Some studies, there've been a few. There was a nice study. This was conducted quite a while ago now, out of Taiwan. This was actually an older aged individual, so I guess this also allows you and I to kind of move into some discussions about aging. What I liked about this particular study, it was all in nursing homes. So going back to that acquisition and exposure and some of the gender associated things. And what they showed was that among the vaccinated men and women, they had more breakthrough cases among the men.
Okay. Were there differences in the vaccine elicited responses in terms of CD8 T cells?
Such a good question. They didn't look, I think all too often in most vaccine studies, everybody looks at antibody and everybody measures it the same way. So, there's a need and a lot of room.
It's easy to measure antibodies. Looking under the street light for your keys. Yeah.
It is.
Happening with SARS-CoV-2 as well, right?
Yes.
People
it is.
Measure mostly antibodies in there.
It is.
Freaking out where, Brienne and I are like, hey, look at T cells.
Yeah, I fully agree with you. But Brianna, I will tell you that we have done work in our flu model, and we do show that females have greater T cell immunity than males after infection. Is where we've shown this most consistently, both CD4 but also CD8, and that included virus specific. And in the paper that's in press right now at PLOS Pathogens, we really show very nicely that testosterone is intimately involved in that downregulation of those virus specific CD8 T cell responses in males.
Cool.
Now, when you age,
I haven't ignored it.
When you age, what happens to your response to vaccines?
Yeah, what happens to
Okay, it goes down.
I knew that.
And here's the thing, though, and I think it's just because we start in a different place, meaning females start in a different place than men. We're showing that if we either completely lose the sex difference in the immune response to the vaccine, or if it's still there, it's greatly diminished. And that impact of aging appears to be greater for females than males. And again, I think it's because we start at a higher place at a younger age than our male counterparts. But it does decline. And I mean, this is where I think it could start to get really interesting with adjuvants. Nobody's thought about do men and women differ in how well an adjuvant would work? Do women even need adjuvants? I mean, this is where I can get on a soapbox about, just precision medicine. And then I do think sex, much like age, should be being considered in the context of maybe not only how we dose, but the formulations.
Somebody has email running. Could you quit the program?
Absolutely. I think it's me. I'm sorry.
No worries.
Done.
You as the guest can do anything you want.
And it was not Coldplay, so it was not welcome.
So did I hear correctly that with aging, the gender differences in reaction to vaccination tend to even out?
Yeah, they do.
How about infection?
For some viruses, for some infections, they even out. For others, they don't.
Okay.
But I will tell you, it's a small body of literature.
Okay.
I think all of you are nodding, and so you recognize what I do. that there's really not been a lot of really good epidemiological studies focused on older aged individuals. Where we do care about older adults is in the context of flu. And so, and so with flu, and I mean, there's some really nice data out of Australia that show this even for seasonal flu, that you do tend to have more severe cases in women in their reproductive years. And that does go back to Kathy's, comment about pregnancy. But then at older ages, it's men.
Yeah.
Is there a difference? Is there a difference in the incidence of shingles in males and females?
That's such a good question. I don't know. I know that's an excellent question. I don't know. The only thing
there you get infected.
I know.
When you're younger. Okay?
I know, but
it doesn't show up until you're older.
I know, and that's really interesting, and I don't know, and I can, so it's not. The data might be there.
Okay.
Because the only thing, the minute you said shingles, when I go through this old Rolodex in my head, I come up that there's a really nice study showing that antibody responses to the shingles vaccine in older adults, it's greater for, even older women than men.
Some of our younger listeners may not know what a Rolodex
is. I know. It's when we used to write everything on pieces of paper.
It occurs to me that one of the challenges in looking at gender differences in older populations is the older you go, the fewer men you have.
There you go.
Group of women.
There you go.
You have an inherent demographic skew that shows up as well.
And so that's exactly what you see. And so I always find it really interesting. And again, I think COVID-19 is kind of interesting like this, that when we go specifically into nursing homes, so COVID-19, we're seeing this male bias in terms of severe outcomes. But when you go into nursing homes, it appears to be female skewed. But what I'm arguing is that what you need to be mindful of is that 2/3 of your residents were females. So you somehow need to look at this relative to who your population was, because if 2/3 of your populations are female and you're getting more COVID-19 deaths in that population, it's kind of relative. It's as much like what we're talking about with race and with African Americans in this country. If, they're roughly 13% of the population, but yet 60% of severe cases, that's disproportionate. There's something there. So I think when we talk about the fact that, like you said, that we do have significantly more women at those older ages, I think that needs to be accounted for.
The other problem with vaccine responses as you age is that memory is also poor, right? It's not just the initial response.
And I don't know if memory responses differ between men and women as we age. And I'm working with a gerontologist at Johns Hopkins, Sean Lang, and we're doing just that. We're really trying to get at more of the depths in people 75 years and older who received the high dose flu vaccine, really getting at characterizing different populations of flu specific memory B cells, kind of going beyond, just that antibody response.
Because there's the classic situation, you make a new flu vaccine, and in August the older people are told to get it, and then by February they have no memory.
No, it's true. And so again, it comes back to, should we be dosing differently? Should we be giving boosters?
Well, clearly, right? Clearly it should not only be different, maybe for sex, I'd like to talk about that, but for age, for sure, it should be different. Maybe they should have different vaccines. And by the way, I've been told I don't have any more bone marrow in my long bones. Is that true?
Really?
Well, you have none?
You gotta have some.
I don't think it's none.
I don't think it's none.
Well,
either.
I like to say none. It sounds impressive.
It does, but it does. It
goes away, right? And is that the same in men and in males and females?
Nobody's to my knowledge, nobody's looked.
All right. What I'm learning from you today is that this is a nascent field, wide open with a lot of not just these associations, but also the hardcore immunology needs to be done.
Oh,
absolutely. At each level,
absolutely.
Right?
I'm getting a whole list of things I want to do.
I love it.
This actually seems like a field that that could use some bioinformatics folks going into it,
absolutely.
especially on the human side with medical records databases. It seems like if you can get past some of the issues with dealing with those types of databases, it could be really useful.
I agree with you.
If you could hack into the life insurance companies,
well,
you'd have it all.
That's true.
Well, you don't need to.
But there are groups at Stanford who are definitely taking some of these, big data systems biology approaches to trying to understand even some of these sex differences. So,
well,
there are pockets of us.
The shingles vaccine. The old one, the previous one, was studied by Kaiser Permanente. They had these hundreds of thousands of patient records, right? Of not only who got the vaccine, but who had shingles then, because they come in. And they can probably stratify it by sex as well. Right?
Yeah.
It's just a gold mine.
Is it known why immunity wanes with age?
Oh my goodness, that's a big question. Yeah, I mean, that is this immunosenescence and what this is. I mean, the short answer is no.
Okay.
I think that's why so much money now goes to the National Institute on Aging, and they have an incredible interest in studies of immunology, I think, for this exact reason. Because we really don't have as good of an understanding of the aging immune system. And I think if we broadly take a life course perspective, it's clear we also don't know at early stages of life. I think textbooks always told us there really wasn't much immunity when we're young. You're relying on your mother, the transfer of immunity from mother to young. But we are starting to understand that even in utero, there is actually development of some immunity in all of us. And so I think that's also really, if you will, in its infancy.
Yes. All right, let's move on to COVID-19.
Okay.
I know I remember back when it was mainly in China already hearing that men were disproportionately affected. So tell us what we know there.
Okay. So I mean, there were two pretty decent reports out of Wuhan, illustrating that severe disease was greater for men. You were getting increased rates of hospitalization, as well as increased death rates. So, of course, I immediately, wrote a letter to The Lancet and an op-ed to a fancy newspaper, and was rejected both places and told it was premature. And then
compared to everything else that's going out there,
I know.
Oh my God!
Don't you love that? I did that like within the first few weeks of March. And you persevere. But then the data started coming out of Europe, and so now Italy, France, Spain, Belgium, Sweden, the UK, and you were still seeing this, and then New York City, as an epicenter, we were still seeing it, and around the United States. And now I can tell you, so there really is global 50-50 is, they're kind of this online nonprofit, and they are tracking all of the what they refer to as sex disaggregated data for COVID-19 in all the countries, and it's this amazing real-time site to go to, and I do think it's changing the face of how we monitor data and consider sex and gender in a pandemic, because there wasn't anything like this in 2009 for the flu pandemic. And now I think it's almost, if you will, shaming countries that hadn't been putting out their data sex disaggregated. You keep seeing regular increases because they can do it. It's just as you all said, even with life insurance, the data are there.
Yes.
There was something in the paper this morning about asking for more demographic data reporting in the United States for all of this. Yeah.
I mean, it is. I mean, it's age. We know age is associated. So the older we are, the more severe disease. Being male is a risk factor. Race is a risk factor. We know that being African American is a risk factor. And you go back, and so when we talk about, staying in my lane with this male bias, I do think it's an interesting intersection of sex and gender. I look at these data and I think it's so profound, this male bias, and it's cutting across cultures. It's cutting across places that may have different societal norms. different, just lots of things that can be different that might be used to explain when we see a male-female difference that isn't upheld across all countries. We might say, well, maybe there's some gender, there's some societal factors in here.
For instance, I remember thinking early on, or hearing early on, that because so many more men smoke in China,
absolutely.
But what it sounds like you're saying is that isn't going to hold up when you start looking at this more global data.
That's exactly. So then you go to Spain, where smoking turns out to be much more equal between men and women, yet the male bias and death from COVID-19 was greater.
Yeah.
So it can't be all smoking. Now, I'm not saying that lifestyle can't affect our biology, so we know that heart disease, hypertension, obesity, diabetes, these can be risk factors for more severe outcomes from COVID-19. And we do know that there are sex-specific differences in the presentations of many of these diseases. So if you have these diseases, it is going to probably impact your immune system and your biology and how well you manage a viral infection. So you can see then there will be some intersection. This is where, so I am working with a lot of wonderful people at Johns Hopkins, developing our own hamster model. And yes, we're considering sex.
So this is the opposite of flu, where
isn't it? Yes.
Right.
Yes.
So do you think testosterone is involved? Sorry.
Well, I mean, we're seeing that play out in real time. There was in this week's issue of Science a nice write-up summarizing some published data out of Italy, as well as some planned trials around the United States, including at Johns Hopkins, because in terms of SARS-CoV-2, that TMPRSS2, that presumed co-receptor is androgen dependent. And there's some data to suggest that men who were receiving an anti-androgen therapy for prostate cancer had lower rates of death from COVID. So I mean, it's correlational, but it's maybe these are some hints that, maybe we're going to be thinking about these hormones, not just the role they're playing in modulating the immune system, but the role that they could also play in modulating receptors that viruses can use to enter cells.
Yeah. I may be extrapolating far here, but given this difference where we seem to see more severe disease in men, does that kind of argue against the idea of this being a lot of immunopathology, since women seem to have the stronger immune response?
Absolutely. But I mean, and there hasn't been enough done. So I have to tell you, I don't know yet about what's going on in terms of immunopathology, and is this virus going to have to make me reevaluate how I've this paradigm that I've developed in my head of how I've hypothesized about the etiology of disease and when we would expect to see a female bias or a male bias. And I think the verdict is still out.
Well, there may also be an effect of the timing of the strongest part of the immune response, right? I mean, maybe women are mounting a stronger immune response early when it's effective against the virus. Men don't get around to mounting it until late when the virus is already on the wane. And
I just thank you.
That's a great point.
Somebody said this. Thank you. And I'll tell you. I mean, it's in preprint, but there was a nice study out of China showing that in families, and so I sort of like that they did family cohorts, where you had known exposures within the family unit, you had a longer duration of shedding and a greater viral load being shed in the men in the household than in the women.
How about the ACE2 receptor? Is that different between males and females?
It does. So we at least it shows higher expression in males.
Okay.
Yeah. I would say where that data is strongest, though, in this moment, as of today, is really it's been in the kidney, because I think that's where it's been more studied, is in the context of hypertension.
Sure.
I think the male bias and ACE2 expression in the lungs, where people have looked, and I wouldn't say they've done a really deep dive into this outside of a little bit of PCR. It's there, but it's not nearly as robust as what seems to occur in the kidney. So, but it could be. It's also X-linked. I can't rationalize, and it's been shown, estrogen has been shown in this case to downregulate expression. So I made that point that hormones can transcriptionally regulate, and it can either increase or decrease expression.
Kathy, you had a question a while ago.
Oh, it was this question that we had from a TWiV, and it seems like Sabra's kind of addressed it, whether if females do have a more robust immune system and clotting problems are a result of a more robust immune system, should females have more microclots and more complications?
Oh, that's a good...
But,
That's an interesting hypothesis. I don't know, but that is interesting.
And I have a question, a kind of more general question.
Yeah.
Are you planning to do experiments to sort of manipulate or address, pose hypotheses and test them, as opposed to data mining of things that comes out?
I think I'm probably a bit more. Oh yeah. So, I think one of the things that I love that we've hit on, I'm very interested in trying to tease apart the sex chromosome versus the sex hormone influences, and are they working together? Are they antagonistic? Are they working in opposite directions, the same directions? One is more profound than the other. And so I have just a fantastic collaborator at UCLA, Art Arnold. And he was a part of a team, and everything is a lot of team efforts, that developed a mouse model called the 4-core genotype mouse model, where they translocated the sex determining gene called SRY, which is on the Y chromosome. They translocated that into the autosome. So now it's not sex linked. So now you can have XX individuals that have SRY and have testes, and you can have XY individuals that are minus SRY, and that bipotential gonad that we talked about develops into an ovary. And so now you can have 4 core groups. You can have XX individuals that have testes, XX individuals that have ovaries, XY individuals that have testes, and XY individuals that have ovaries. And now you can kind of start to really get at mechanistically what's mediating some of the sex differences that we're seeing. Both in, responses to vaccines. So that's where I have money. But you could also use this to try to also tease apart the pathogenesis of infection as well.
And so those are going to be for virus models where you can actually infect those mice. And my question was a little bit more going toward the SARS-CoV-2. If there's any way that you're going to be able to do manipulations or things like that in collaborations.
Absolutely. So I'm trying. So, yes, I think, Stanley Perlman, is, friend and colleague, and has been a believer in these sex differences and published on sex differences, in SARS-CoV, in a mouse model. So he and I are in a lot of discussions about some of these mouse models. And,
I think, didn't you write an article that was in the New York Times op-ed page together? Or you were mentioned.
Oh, you're so
something.
kind.
Yes.
No. So after being rejected, thank you. And I did write my op-ed with Stan.
Right.
Then the reporters came to us, and as this was developing, so we got lots of quotes in a beautifully written article that was not an op-ed that we wrote, but he and I did write a beautifully rejected.
Beautifully rejected. So we have focused on disease in COVID-19, but is there any evidence that actual infection rates differ between men and women?
So far, that's a little bit more heterogeneous. By and large, no. By and large, we're as if I take a broad brushstroke, we're seeing that cases are pretty consistent between men and women. Now, for some countries, there's a female bias, and for others, there's a male bias. And I think that gets back to that gender associated, who's getting exposed.
And who's getting tested.
And who's getting tested and who has access to testing. I mean, so, I realize in the United States, we don't worry that as women, we may not have access, but in other countries, the women do not have access. And so do you see a male bias in cases because there's just more testing of men? So this gets a little sticky for truly analyzing and maybe interpreting, I should say.
Got it.
This is gender at the cultural level. I mean,
yes.
I was driving in this morning and thinking about our conversation, and there are people cutting grass. They're all guys. They're construction. And they're in their face. They're yelling at each other. And I said, women wouldn't do that.
That's called risk-taking behavior. Yes. And I will tell you, so, I assume all of you know the Darwin Awards.
Yes.
Okay. So people have analyzed, and their published data showing that roughly, between 70 and 80% of all Darwin Awards are won by men. Okay. It's
good at something.
You take more risks. Yeah. Sometimes that's probably a good thing, but then there's the other time.
Got it. You also wrote an opinion in JCI that the vaccine trials should be done differently. Tell us about that.
So that opinion piece was, I think whether it's vaccine trials or I would argue drug trials, that we need to be not just considering balancing sex of these clinical trials, meaning not just the inclusion piece that women should be included, but that we need to be comparing outcome data to see if we're responding differently. There are data, and this is true for both vaccines as well as for lots of different drugs. Women are significantly more likely to report adverse events than are men. And I've received every response to this under the sun, ranging from, that we complain more, to the more positive gender associated response to me that maybe we're more in touch with our bodies and how we feel. And, but I also, I think there are data showing that our livers process things differ. We process drugs differently. We actually don't clear things as quickly. Go to the CDC website and see why they recommend less alcohol for us. There's some biology behind some of this. And so, going back to the vaccines and those, we're only measuring antibody responses, that we should be measuring T cells, especially with some of these, I think RNA-based vaccines, even adenovirus-based vaccines. I think they need to be considering this.
And you mentioned antivirals as well, right? The responses could differ.
We're using immunotherapies. There's ample data out there showing that there are male-female differences in responses to immunotherapies for cancer, but yet that then doesn't translate into how we analyze data.
Yeah. But can you envision a time now in the future with this vaccine or another where what kind of data would you need to justify, say, doing different things for men and women, even a different vaccine perhaps?
So I think where we're at today is, I got to take a step back. Today, I think we are to the point where I think it is irresponsible that anyone designing clinical trials is not acknowledging that they should have an a priori hypothesis that males and females will differ, and that needs to be built into the statistical model, and that it should never be done. It should not be done anymore as a post hoc analysis that is not considered valid because a study was not powered well enough to do this. So I want to start there. I think simply, because that's where we're at. And I think if we could get that so that we could have the correct trials being done, then I think we could start to get to a place where
that is not where we are.
No.
And just to back up for the listeners that might not know what it means to be powered.
Thank you.
Just means really having enough sample size, having enough of both and women.
Right. If you had 1,000 people in your trial and only 100 of them were female, it is not adequately powered to look at gender differences.
Absolutely.
But it seems like what you would need to make this happen, since the folks funding the big clinical trials are the companies who need FDA approval, is for the FDA to do what the NIH has done and say, this needs to be in the data if you want to submit for approval.
Yeah, that's why I was confused, because we had discussed earlier how the NIH has incorporated this into a lot of stuff, but for trials, we need the FDA to buy in the same.
The NIH stuff is only for even only for preclinical trials.
Okay,
and the FDA, they have their Office of Women's Health. They push for this. So there are efforts, but they often have to rely on what happens at the NIH to drive what will happen for them, and that has the NIH put forth their policy. They announced it in 2015. It went into effect in 2016. The FDA still has not.
Well, what kind of difference do you think would warrant having even a different vaccine for men and women? How big would it have to be? I know it's a tough question, but
it's such a tough question because, especially for something like this, where we don't know what is going to be that correlate of protection, what is going to be that meaningful response that tells us something about protection.
We have a rich set of data, though, for measles vaccine. Is there anything that emerged from that?
That is the good question, and yes, and I'm at the top of my head. I'm not summoning the direction of that difference. And there is a group led by Katie Flanagan, who is a dear friend and collaborator in Australia. She does a lot of work with sex differences and vaccine-induced immunity in kids under the age of 5. So she deals exclusively with.
And then there's rates of malnutrition differences in stressed populations. That
absolutely.
Mostly the males get the land share, unfortunately.
That's all true. I mean, and there you get into the gender. That's horrible. Gender. I mean, there are studies showing that respiratory infections in kids differ in some countries, and it was linked to and earlier, some of the unusual. Let me start all over. Males are typically more susceptible to respiratory infections early in life. So when you want to know who's got either a viral or bacterial pneumonia in a pediatric population, it's the baby boys. But in some countries, it's girls, and it comes back to who's being breastfed and who's getting the nutrition. So it goes back to what I was saying to you when you see that there's a flip in the male-female difference, depending on the country. Maybe you need to look to some of these gender-associated factors that might be impacting and masking any of the biology.
To go back to Vincent's point, is there a root difference that might emerge that would warrant giving the vaccine another way to females and to males?
I love it. I think that's really interesting. I will tell you, for the flu vaccine, we really see this robust difference with the intramuscular vaccine. I've not seen data reporting comparing males and females for flu myths, so I can't really speak to intranasal. But I will tell you that there was a nice intradermal. So it was a smaller needle that was being used to give. And if you gave it intradermal, you eliminated the sex difference.
Would that apply to the microneedle patch too?
Yes, I don't know, but I'm hypothesizing maybe. So
why would that be?
Nobody's, including myself, nobody's really gone to see, well, are we driving differential responses? Are you getting the skin? That's exactly what I was thinking. Right. And maybe males do that well. I mean, maybe with wounds and wound healing. Maybe
you have to be doing something.
Exactly. You do a lot of things.
It may also be that the transdermal or whatever is just inherently inducing a stronger response.
Absolutely.
So maybe you've already hit the ceiling for the women, and now you finally hit that same threshold for the men.
So this is where I think we need to be going, because I think ultimately the goal is to protect us equally.
That's right.
That's the ultimate goal. It's to eliminate some of these disparities.
Well, my understanding is that microneedle patch would replace all needles, which could be a good thing. Maybe it would balance everything.
Eliminate the sex difference.
That's cool. Sabra, is there anything we have missed that's really important that we should have talked about?
This has been lovely.
Okay.
I can't think of anything, and I'm a little brain dead.
I have a question for you. What's the origin of your first name?
Oh, I know what that is. I know what that is.
Okay.
It's a beautiful name.
Well, thank you. So it is Israeli, and it by definition means it's the feminine form of native born in Israel. So now I get to announce to your listeners who know that I think I'm Hawaiian, that I am a fake Sabra. I am not a native born in Israel. And I can't go to a conference and not be asked, are you a real Sabra? To which I must say no. But the other, it's also a fruit on a desert cactus that's prickly on the outside, tender on the inside. And that is how my mother describes it.
And do you make your own hummus?
I do not, but the hummus helped with everybody knowing how to pronounce my name.
Exactly.
No, I asked Amy in my lab, what does this mean, Sabra? And she told me out of Israel. So
yeah,
but you're
which I am not.
It's okay. What's out of Hawaii? Who knows?
Sabra, you're going to have to listen back to this and take notes. I think there's about 5 or 6 grants.
Yes. Oh, I hope so. I hope so. I mean, perseverance has been the name of my game, going back to being a mutt, and trying to figure out where are people interested in this and see this as a priority. And I think COVID-19 is putting this on the map in a way that, I wasn't, I was prepared for. It's what I do, but it's been exciting to see such an interest in this. And
well, the fact that it's killing males at twice the rate of females may get some attention.
Yes, leadership cares.
Yes.
Right.
Hopefully our listeners will also hear all of this and start actually changing some of their experimental design or taking things into account in experimental design.
Thank you. I hope so. It's not that I think everybody's got to study this, but the reporting, the consideration, I think it's important.
Yeah.
We always have to ask.
I have to say, when I started doing mouse experiments in the early 90s, I automatically would do half males and half females. I said, why wouldn't you?
Pretty good.
And I didn't know anything. I didn't know anything.
Well, you knew something.
Wow. I was raised.
You were ahead of your time.
Maybe I was raised well in my family.
You must have been. Yeah.
Who knows? I can blame my parents. Before we end up, Kathy, want to say something about ASV virtual workshops?
Sure. So the virtual workshops are going to start in the week of June 15th, 19th. These are replacing the canceled ASV 2020 meeting that would have been in Fort Collins. And these virtual workshops are a grassroots effort by a bunch of virologists from ASV, shouting out to Stephanie Karst, the program chair, and the ASV administrator, Andrea Garcia, and our office helper, Haley Goran, who's been helping keep the webpage up to date. So if you go to www.asv.org, on the main page, there's a big purple button to click about the virtual workshops. That takes you to a page where soon, if not already by the close of business today, there will be a Zoom link for each of the individual workshops. Anybody can register for those, but you do need to register in advance by clicking on that Zoom link. And it's going to be people who had submitted abstracts to ASV for the 2020 meeting, and people have volunteered a lot of time to do this hosting and so forth. And so, as I said last week, the ASV office didn't have the bandwidth at the time to try and do anything bigger than this. So we're really grateful that people have stepped up and done this. And I think it's going to be really exciting. Just like a regular ASV meeting, there are overlapping workshops. And so you have to pick and choose which ones you want to go to.
But this time you could have multiple screens.
You could.
That's
you
true.
could.
Yes, that's right.
So,
Kathy, a couple of questions. Can I register if I didn't submit an abstract?
Oh, of course. You don't have to be an ASV member. You don't have to have anything at all. So yes, there's a poxvirus workshop. You can go to that.
Yeah, I got an email from some poxy friends with the link in it. So is there a window of time during which I have to register?
We're trying to encourage people to do it, at least an hour or two before the scheduled time, so that the host.
Can I do it now?
Of course.
Okay.
I've already registered for one. Yeah,
Wait till we're done with TWiV content.
The ones that I'm involved with, Rich, I will say that depending on how the host set them up, there are different maximum numbers that people can have. I mean, they're relatively large maximum numbers either way, but it is theoretically possible.
There may be a limit.
If the global virology community really jumps onto this, there could be some workshops that sell out, as it were. So,
I mean, you may want to do this all the time, just considering how many people can't afford to travel. They've got their computer and they can come on board. So you might be onto something here.
Right.
Even internationally.
Right.
Yeah, absolutely.
Yeah, sure.
Yeah.
All right.
Well, then we have a lot of time zone issues. So you'll see that we've listed Eastern time for everything. And then oftentimes there's something underneath, and that's usually the time zone of the host.
Oh, do you, may I make a recommendation? Could you also list UTC?
Well, they can,
never mind. Next time.
they can figure it out.
That way everybody can convert to their own time zone.
But pretty much everybody with a phone can type in a city and figure out New York is Eastern time, and type in New York and figure out how it is.
The standard for dealing across time zones is because I frequently am calling people, in other countries, and I always put the GMT time of the call in parentheses just so we know that's right. Because I don't keep track of who does daylight saving when.
Right. Well, and that's why we took out the saving and daylight. The standard and daylight, because people were saying Eastern Standard Time, and they really meant.
They don't mean. I get that all the time. Yes.
They just mean Eastern Time.
Yes.
So,
All right. Thanks, Kathy. All right. That's TWiV 624. You can find the show notes at microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. If you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. And depending on how much you give, you can get a mug like this one here, right? And I'm way back on all the things that I have to send to people, mugs and T-shirts and so forth, but be patient, please. Things have changed a bit. Our guest today from Johns Hopkins Medical Center, Sabra Klein. Thanks so much for joining us. It was really
thank you so much. This was so meaningful for me. Thank you.
Good. Dixon Despommier is at trichinella.org and thelivingliver.org. Thank you, Dixon, and happy birthday again.
Thank you. This was a great session. I truly enjoyed it.
It's a good 80th birthday session, right, Dixon?
That's a great way to look at this. Thank you so much.
I'm glad that you decided.
The rest of my life. How long will that be? I don't know.
Not long.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Happy birthday, Dixon. Thank you, Sabra.
Thank you.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. Happy birthday to our octogenarian. And thanks, Sabra. It was really fun to have you.
It's getting older already.
Brianne Barker's at Drew University on Twitter, BioProf Barker. Thanks, Brianne.
Thank you so much. It was really great to be here. Happy birthday, Dixon and Sabra. Thank you so much. I've learned a ton, and we'll be updating my immunology lecture even more than I did last time I heard you talk.
Thank you.
Alan Dove is at turbidplaque.com on Twitter. He's Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure. Sabra, always great hearing you. And Dixon, just, keep going for another 80.
I'm going to give it a good shot.
Dixon, I want to say that you are not the oldest podcaster. I have that honor. Elio Schechter on TWiM is 93.
Whoa.
And he's sharp as ever, so you can do it too, Dixon.
All right.
I'm not discouraged by that. I think I'm going to give it a shot.
All right, very good. I'm Vincent Racaniello. You can find me at virology.blog. I want to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Hey, Dixon, what are you going to do? How are you going to celebrate your birthday?
I'm going to make
He just did
a Chinese meal.
Oh, yeah?
We're going to have some shrimp. We're going to have some baby bok choy, hot and sour soup, a little beer, and a wonderful dessert made by Bobby Flay called a banana. A banana chocolate cake or something like that. It's a loaf cake, and it's fantastic. It's already
got a recipe I got to send you. It's a breakfast recipe called something like spiced bread, egg in a hole.
Let's do that.
So it's a big thick slice of sourdough or something like that, and you cut a hole in it.
Right, and you put the egg in the middle.
You make a slurry.
Sure, the faces. There's a gender difference here. Okay, you've got to writing down. What is that called again? Yeah. I think I saw drool on a few of the faces, and it wasn't the females.
Now wait a minute. You make a slurry of olive oil and turmeric and coriander and a little bit of red pepper in a pan, and then you fry the bread and the hole in it, and you stick an egg in it and flip it. It's amazing.
I'm anxiously waiting for your recipe.
I'll send it.
Yeah, and photos too.